You mentioned something interesting just now. You talked about the blood type diet. How true is it?
It was crazy useful.
What is aging? What's the definition?
Like our birth age, right? Chronological age, your biological age. You'd have functional age, like are you faster, stronger, better grip strength, you name it. Perceived age, right? So that's, how old do you feel?
So theoretically, if we don't have these senescent cells, can humans live way beyond 100 years?
But it's the quality of life, right? Like, can you perform better at whatever your age is?
All 8 billion of us are doing metabolism at all times. This show is about learning what metabolism is, how it affects you in every way possible, from mood and mental state to performance and energy. We are all about fine-tuning the human experience for you to achieve the best self you can be. And if you are someone who loves science, curious to know how your body works and how to optimize it, then you are in the right place. This is the H.V.M.N. podcast. In this episode, we have Dr. Gregory Kelly, who is the Director of Product Development at Neurohacker Collective, naturopathic physician and author of the book Shapeshift. He was the editor of the journal Alternative Medicine Review and has been an instructor at the University of Bridgeport in the College of Naturopathic Medicine, where he taught classes in advanced clinical nutrition, counseling skills, and doctor-patient relationships. Dr. Kelly has published hundreds of articles on natural medicine and nutrition, contributed 3 chapters to the textbook of natural medicine, and has more than 30 journal articles indexed on PubMed. His areas of expertise include nootropics, anti-aging, regenerative medicine, weight management, sleep, and the chronobiology of performance and health. If you are curious what are the different types of aging definitions, what is senescence and senolytics, as well as how to live a longer, healthier life, then this episode is for you. So stay tuned and enjoy. Welcome, Dr. Gregory Kelly, to the Health via Modern Nutrition podcast. Very, very nice to have you here. Welcome.
Thanks for having me this afternoon. I look forward to talking to you and sharing with your audience.
No, absolutely. I would love to, can't wait to pick your brain around your expertise and knowledge around aging, senescence, and all of that. But before we jump right into that, could you please introduce yourself? How do you get into this area? What do you do? Let our audience know you a little bit more.
Sure. So I'm a naturopathic doctor by profession. Graduated in '96 from, at the time, it was the brand new school. It was called Southwest College of Naturopathic Medicine. And worked for a company called Thorne, one of the biggies in the supplement space, both as a student rep and then immediately after. But back in those days, Thorne was doctor only. They didn't sell directly to the public. And so my role during that year was largely to educate doctors and answer their questions. And then after a year doing that, I went out and practiced for a bit and saw patients and actually worked in a clinic with Dr. Diadamo, of blood type diet fame. He met me when I worked for Thorne and basically said something like, "Oh, I don't like many people. I seem to like you. What do you think about moving to Connecticut?" So at the time I was thinking, how can I see a lot of new patients quickly? And that was an ideal situation. And then I've been, done things in corporate wellness, dietary supplement industry, and most recently I'm the director of product development at a company called Neurohacker Collective. And we tend to do quite a bit of our products and focus is either on the brain or longevity. So those would be 2 passionate areas for me.
Great. I mean, we all love the brain. We all love the longevity over here. I think a lot of our audience are also people who are trying really hard to either change their lifestyle to live a healthier lifestyle, or already practicing a healthy lifestyle, but want to know more to optimize their current lifestyle so that they can live longer, healthier, move faster, quicker, and be more stable as they age. So you mentioned something interesting just now. You talked about the blood type diet. So let's talk about that for a little bit. How true is it? Like, I've heard it doesn't even matter what blood type you are. I mean, it's pretty much debunked now, no?
So what I would say is it was crazy useful. So I tend not, things start to get mushy pretty quickly when you start to get into scientific rigmarole. But the way it would functionally work would be, so I'm a blood type A, but I also know that my dad was a blood type O. So I would be an AO, right? I would have inherited an A from my mom, an O from my dad, and the O is recessive. So A is dominant. And when I was born, I wouldn't have had any antibodies against blood type B or blood type O. What we develop those, our immune system learns. And one of the things it learns from is food. So foods have things that are antigenic, right? That would cause our immune system to respond. And that's pretty well known. So the idea behind the blood type diet, and I'm going to probably misspeak some of this compared to how Dr. Diademo would, goes back actually to his father. So Peter's a second generation naturopathic doctor. His dad was more the mad scientist, like in practice, that had all kinds of people come to his office, which at the time was in Brooklyn, New York, I believe, and had this crazy idea like, oh, food carries nutrients, or I'm sorry, blood carries nutrients, food is nutrients. I'll tell people to eat different things and see blood type everyone and just see who does well on certain things and not so well at other. So it was very much an N of one taken to like an extreme. And what he noticed over time were these patterns, like, oh, if I tell O's not to eat these things, but eat these, they tend to do better. Where if I tell A's to do that same thing, they tend to do worse and do better if they do these. And so what Peter did was try to then put some scientific theory why it worked. Mm-hmm. But the observations that it was useful predated Peter. Does that make sense?
Yeah.
And I had, and you would see this, right? Like you have anyone change their diet dramatically, there's a subset of people that are going to respond really well. But there was people I saw with crazy, hard to treat, rare conditions that on their own had read his book, followed it, and came in just to almost tell us, like, wow, this worked for me. So hence my terminology, it seems useful.
Yeah, it seems useful. There's no any strong evidence except for anecdotal, I believe.
Correct.
But then again, we talked about this. I interviewed Dr. Daniel Pompa before, and he talked about the differences in diets. When you change the diet, you create some sort of stress system where your microbiome will adapt to the stress, and some people, because of that stress, sort of fare better compared to the rest. So we don't know.
Yeah, the other thing I think that with blood type, like two other points before we move on to something else, would be that those things that make my blood type A aren't just on my blood cells. Mm-hmm. They're literally everywhere. Anywhere that interacts with the outside world, our mucus, things like that, we secrete that blood type antigen. So I came from the Navy before becoming a doctor, and anti-submarine warfare was what my ship types always were. You would have what they would call chaff, right? So you would like deploy these things to act as a decoy. So blood type, when we secrete those antigens into, say, mucus, it acts as a decoy. There's things in the bacterial world that bind to that. So our blood type codes a lot of our susceptibility to what can and can't interact well with us in the bacterial world. And a lot of those same things that would make something an A or an O antigen are basically long sugar or amino sugars, glycoproteins, and those are food for our gut microbiota. So no matter what we consume, our blood type actually will dictate some of our gut microbiota, just like other aspects of our genes would. So there is like science there, but it's probably not as black and white as it would be made out in the book.
Yeah, and I think with the interest or the growing interest in microbiome and gut health these days, probably a lot more funding may be allocated to studies to specifically look at these and actually answer these questions from a very objective scientific point of view. So let's talk about your passion and your area of expertise. What are the main theories of aging? Let's go back to basic, tell our audience, what is aging? What's the definition from a systemic level, cellular level? What are the main theories?
Well, so I think when we think about aging, sometimes you'll see things like our birth age, right? Chronological age. And I think for all of us, me included, but probably most of the listeners, we want to be younger than that in a sense, right? We want to perform at a better level. So that's things like now your biological age, which, those would be like the DNA methylation test or other things like that. You'd have functional age, which now you get into like, how are you function? Like, are you faster, stronger, better grip strength, you name it, than your age dictates. Perceived age, right? So that's, how old do you feel? And most of us are going to say, oh, younger than our chronological age. That's felt age. And then perceived age would be how old someone, like really a group of people looking at us, would guess we are. And so the goal for me always is we want all those ideally to be, younger than our biological age. And in terms of how then science understands it, there's almost then two camps of like why we age. One would be damage accumulation. So everything's lumped into, well, we age because damage occurs in our cells, mitochondria, and that accumulates. And that accumulated damage over time causes poor function, and that's what aging is, right? It's just an accumulation of damage. And spinning out of that is the solution is let's, get rid of or repair that damage. The other camp is more about what's called programmed aging. And think of it almost like software, like it's designed to get us past a certain point so we can reproduce. And after that, the things that were advantageous for that are actually programmed to kind of fall apart because we don't need to do that anymore. So that the software runs differently as we get older. And while there's these two camps, there's definitely overlap. So we're going to talk about cellular senescence. You could make a strong argument it's the damage control, but it also looks like we build more senescent cells, like almost as a program as we get older as well. So it's probably not black and white. No matter what the theory is, it just explains part of it. And I'm always most attracted, well, is there something we can do? And the damage control camp seems like there's more we can do currently than trying to change the programming.
And let's explain what is senescence, to those who are not familiar with that term.
Senescence has to do with, so think of a cell as just like being born, living its life, and then passing away. Now there's scientific terms that get tagged with that, and the passing away would be apoptosis, cellular death, and more specifically like a planned cellular death. An unplanned one from trauma, that would be necrosis, right? So, but what happens in a cell's lifestyle or life cycle is that cells can be stressed just like we can be stressed, and they'll try to respond and adapt to that stress. And I tend to put that on a continuum. So at a low level of stress, something they can adapt to, you'll see them upregulate things like antioxidant defenses, right? They'll make more glutathione, as an example, but enzymes like SOD and catalase that help them deal with that oxidative stress. And no matter what the stress is, oxidative stress is almost invariably a side effect. So bottom line is with that modest amount of stress, they'll try to toughen up and defend themselves. And if stress is beyond that, I go back again to like when I was in the Navy, right? You're going to take some damage, like you're hit with something, and you're going to try to clean up the damage, right? So in the Navy, we would have had like a damage control party, right? That was their job, like go to wherever the damage is and repair. And something like autophagy would be in that, right? Which is autophagy is specifically recycling of damaged proteins, organelles, things inside a cell, to try to reuse those things and repair the damage. But if stress is beyond that, then a cell can become a senescent. And what that means is the damage is beyond where it can be repaired, and so the cell's kind of targeted to go through apoptosis. And once that happens, it's not allowed to make new copies of itself because we don't want to reproduce damaged cells. So senescence would be that middle ground between, okay, there's beyond what I can repair, but I haven't gone through apoptosis. I haven't passed away yet. And so a senescent cell, by definition, is that. And why that's important is these senescent cells accumulate in our tissues and organs as we get older, which is relatively newish science, meaning, it goes back 20 years or less. But then the contribution to how that, causes our tissues to perform poorly as we age, that's really still exploding. Like I would say most of the science on that's been within the last 8 to 10 years. So just in a simple thing, sense of like the analogy we'll often use at Neurohacker is think of a plant. young, healthy plant, all the leaves green, plant is thriving. But maybe because of nutritional stress or not enough water or pests in the environment, some stressor, now one or two of the leaves starts to turn yellow. Ideally, that leaf will just fall off, right? And apoptosis, that word we use, that comes from a Greek word that literally translates as falling off, right? It means the same thing. It's this idea of, oh, the cell's not healthy, it's done its job, it falls off. What happens as we age is those yellow leaves, more and more of them pop out on the plant. And once a plant gets to a certain amount of yellow leaves, it will struggle to stay alive, right, to do its job, because the yellow leaves are still sucking resources, right? They're still using nutrients. They're attracting now pests and other things from the environment to cause almost an accelerating cascade of damage. So think of senescent cells as the equivalent. It's these yellowing leaves equivalent in our tissues that are just growing and spreading more and more as we age.
So would you say senescence is essentially a adaptation process to try and recover, but ultimately not able to?
Yeah, I mean it's a normal cellular response, right? So
I'm just thinking it from an evolutionary point of view. What's the point of senescence when you reach a certain threshold of oxidative stress, for example? Why don't you go straight to apoptosis, which is plant program death, and you send signals and say, hey, like these cells are not getting enough nutrients, so it's better to just program their death. Why bother with that intermediate period?
A couple of reasons. So one of the things senescent cells do is they send out information into the microenvironment around them. So cytokines, other immune markers, and they kind of call in the troops. So like, oh, there's a problem here. Like we need immune backup to come in. So like most things in physiology, it's not this is good, this is bad. The context matters, right? So in like pregnancy, as an example, like a developing fetus, senescent cells are part of that. They do an important job at tuning things so that the fetus develops correctly. In a 20-year-old that just runs a marathon, will create some senescent cells as part of recovery, right? Because they inflicted some stress, some trauma on their tissues. The difference between that and when we think of senescent cells and aging, I term the difference between transient and lingering. So it's completely natural that we would make some transient senescent cells, that they would do their job, and that they would either go through apoptosis, so the immune system would find them and gobble them up. What happens in aging is they don't stay transient. They accrue, right? They become these lingering ones. And the other thing, I think that part of the reason, in addition to that they can do something useful when they're transient, is that too much apoptosis all at once can almost overburden the immune system. Like there's concerns too much apoptosis may drive something like autoimmunity. So senescence does its thing. It's just like most things with aging, when something gets so far out of balance, then instead of doing its job and contributing to health, now it's impeding it.
Those are great explanations, by the way. I love it. It reminds me almost of inflammation. Right, you get the acute inflammation and chronic inflammation, and a lot of people, they were talking about how inflammation is associated with chronic diseases, but inflammation in and of itself is a defense mechanism, and it essentially generate it when you do exercise, when you have stress and whatnot. You will upregulate inflammation, and that is just a coping mechanism at which your body is trying to cope with the stress. However, when you accumulate chronic inflammation with all the signaling molecules running around the body, it's what's causing the chronic diseases. So that's the difference, and that's also how you explain senescence, and that's great.
It would be very similar. Like you could even, instead of the transient and lingering, you could use acute and chronic and still be on the money.
Yeah. So as we were talking about senescence, there's also another term I would like you to explain, which is senolytics. What are senolytics?
Yeah, so in 2015, researchers at Mayo Clinic and at Scripps Institute of Aging, for the first time, found that there was natural compounds. One was from a plant, one was a medication, that if you essentially put them in a cell culture with senescent cells, they would cause those cells finally to go through apoptosis. They would normalize them so that they would fall away. And they used the term senolytic to tag things that could do that. Quercetin was one of the two at the time that they did. And what they did is they looked at mechanisms. So there's a couple commonalities that senescent cells all share. Think of senescent cells as a category as opposed to a single thing. But I mean, within that, they resist apoptosis, so they resist this falling off process, and they secrete these compounds into their microenvironment, would be two of the main things. And the third is that they can't replicate, right? They don't make copies of themselves. And so what those scientists did is they tried to find compounds that would work in those anti-apoptotic pathways in cells to toggle it, so to speak, so that instead of resisting, they'd be like, oh, cool. Like, I'm happy to go through apoptosis now. And so looking at those mechanisms, and some of them have to do with nutrient sensing, cells responding to nutrient stress. They identified a couple things, tested a few, and like, oh, quercetin seems to work. And this, medication called dasatinib seems to work. This is pretty cool. What happens now if we give those to animals that have a bunch of senescent cells? And what they then found is like, oh, in these tissues, senescent cells numbers drop down, and that organ performed better, or that tissue performed better. And lo and behold, the mouse, rat, seemed to be healthier than they were before we did that. So senolytics became the catch-all term for things that would fundamentally help correct that resistance to apoptosis, so that these cells would finally fall off, and the immune system can then come and scavenge the debris and put it back to good use. And once in a while, I know you look like you have a question. I'll get asked, like, how do, all right, so we're detoxing these cells. How do they, how do we get rid of them? Are they going out the lungs? And they're not, right? So once a cell goes through apoptosis, think of it exactly like that leaf falling off a tree into the soil. It's now going to be recycled and used for new growth. Our immune system does the same. Once it scavenges debris from cells, it recycles and puts it back to use.
So a moment ago, you were saying if too much apoptosis happen, you might trigger autoimmune, right? But then senolytics essentially almost accelerates apoptosis and that process. Wouldn't that be harmful, or am I misunderstanding this?
No, I think that's a great question. And it's why from the get-go, those research scientists came up with this idea, well, let's do what they called hit and run dosing. Let's just do this for a cycle, 2 to 3 days at a time, so we don't overburden things, and then give a window of time where we're not applying senolytics, so that we don't, by mistake, inflict damage. So it has to do with really the dosing protocol from the get-go of senolytics is this idea of cyclic dosing, but hit and run dosing would be how the original scientists described it.
Okay, yeah, that's definitely a question I want to ask around the dosing. But before we get there, I want to ask you from a scientific point of view, do we, as of now, know if there is the difference between the threshold of different cells in our bodies to reach senescence and to reach apoptosis? Do we know what sort of markers they are and how much before those events are triggered?
We really don't. And so most of the work on senescent cells, because they're hard to measure, even a scientist working with them in cell cultures needs to often look for a couple different markers all at once to be sure, like surface markers, the way it stains, things like that. But going back, so I guess historically, the idea of senescence came out of telomere attrition, like shortening telomeres. And what they found, this goes back to Hayflick and his work in the early 60s, but that cells would only reproduce so many times in a cell culture. It's roughly 50. And then they would just stop reproducing and go into this senescence state. And so early on, by the 70s, you had this idea, this probably has something to do with aging, right? Maybe aging is just that we run out of ability to make new cells, which that's not actually true. We maybe in some, tissues or some contexts, but basically that wouldn't be the driving thing. But what they also found is what is called premature stress-induced senescence, way before the Hayflick limit, telomeres are still plenty long enough. If you do things to stress what's in that culture, right? Ionizing radiation, UV light, lack of nutrients, you name it, you can cause senescent cells to essentially have enough damage that they'll become senescent, with telomeres being fine, DNA maybe being fine as well. So my bias always when it comes to like these stress induced things is a game that my siblings and I had in the late 60s. So I'm dating myself, but it was called The Last Straw. And just for the audience, it was this plastic camo with two buckets, one on either side. And there was different colored straws. Some were thicker, some were thinner, right? So they weighed differently. And each turn would spin a wheel. And if, mine was blue, I had to put a blue straw in. And at some point, you'd put the last straw in and it would cause the back to collapse and your team lost, right? So when I think of stress, that's, I think, a useful analogy, right? Like we all are like a camel, right? We can carry so many straws, right? We have some degree of resilience. We can do things to try to build more. We all have certain straws, some we share, right? Our environment has some straws that there's not much we can do about. And then the way we live, our past, you name it, right? Those could be other straws. And we never know how close we are to that last straw. We don't, right? But what you see with health challenges, what I would have seen in practice is people could routinely after the fact come in and say, oh, Dr. Greg, like this happened, and then my health, like it's never been the same since, right? Whatever this might be, right? A toxin, a infection, a vaccine, like change in diet, you name it, right? Mental, emotional stress. So the goal has for me always been the same. Like what straws can we identify and act on, right? Let's do our best to either get those out of the basket now or make a plan to get those reduced, right? And what can we meanwhile do to build resilience, right? To make your back so you can carry more straws. Because none of us ever want to get to that point of the back collapsing. And so kind of the long story of how I wove there, you had asked about, do we know exactly what would cause, like this cell to be, instead of maybe going through apoptosis, going only senescent? And I would say no, right? Like we know like the same category of stress applied in different amounts might cause some cells in again, a controlled experiment to go through this apoptosis. Other ones would only have become senescent. Other ones maybe only would have needed to do autophagy, right? They would have been more resilient. So to me, the answer is not like, what's the stress? It's, is there something we can do to minimize it? And how can we be more resilient if it happens? Because the if often turns not into an if, but a when.
Right. So essentially what I'm hearing also is by addressing senescence, by using senolytics, for example, and by really preventing the accumulation of senescent cells, we are essentially aging. We're not aging less, but we're aging with less quality prohibiting illnesses or diseases or
Yeah. So I think a lot of health is a numbers game, for lack of a better way to describe it, right? Like it's about staying below a certain threshold of dysfunction, right? So that an old naturopathic principle would be the health of our tissues is reliant on the health of the cells in those tissues. But again, going back to that analogy of a green plant, as long as we keep the threshold of damaged leaves, like we prune off the damage, that plant will do well, right? As long as the other resources are there. It's only when you allow the yellow leaves to accrue past a certain tipping point that now the whole plant's health is in jeopardy. So when I think of senescent cells, I think of them that same way, right? Where they're not easy to measure, right? You'd have to really do a biopsy, which not many people are signing up for, to know what's in tissues. But based off animal studies, we do know with certainty that they gradually accumulate over time. So an old, like baboon, as an example, may have 20% of their cells in most tissues. Some tissues more, some less, would be senescent. One that's 10 years like younger, equivalent, like human equivalent, would have less, right? Equivalent, 20-year-old would have, vanishingly small numbers. So we know these accumulate, and when I think of it, it's more like, okay, what can we do that's the equivalent of pruning them away? And that's what the promise of senolytics is. They act like the gardener, right? They periodically prune these away so that we stay below that threshold, ideally. And if we're already above it and experiencing some kind of dysfunction because of that affected tissue, then maybe that we dip now below enough that those things also improve symptomatically.
I get to interview all these doctors, scientists, and cool people in this health and fitness industry, all made possible because of this podcast that is funded by the company I work for, which is Health via Modern Nutrition, or H.V.M.N. And it is not that they pay me to do this, but I genuinely love and believe in the product Ketone-IQ. I use it every day before my podcast, before my workout, or even after my workout for recovery. There hasn't been a single supplement that can give me such a drastic change in subjective feel within minutes as much as Ketone-IQ has. For those of you who do not know me, I'm from Malaysia, I got my PhD from the UK, and my passion is in science and chronic diseases. And I believe it is all about transparency, scientific integrity, and about sharing with everyone so that everyone can benefit from it. And if you like this content and our work, please do support us by liking, leaving a review, or sharing with your friends and families, or even buying a shot of Ketone-IQ at any Sprouts nationwide in the US, and the first shot is on us. Just scan the QR code and you'll get your money back for your first shot. You can also use the code H.V.M.N. POD20, that is H.V.M.N. POD 20, and get 20% off your first purchase at the H.V.M.N. website. Now, I'm just thinking of a hypothetical situation here where someone is accumulating senescent cells due to accrued stress, right? And these senescent cells were programmed to enter the stage in order to rectify or recover from that damage. If you use senolytic to reduce the senescent cells, would that interfere with the natural process of defense mechanism? Would they need to also not just take the supplement, but also really address the root cause that is causing the senescence in the first place?
Yeah, so let's answer that in two starts. So let's start with what's causing like the accumulation of more and more senescent cells. And that's probably like a combination of four different things. One, it seems like genetically we're just programmed to make more of them with the passing of time, right? So there's that's where the programmed aging piece fits in, because otherwise these would be clearly damaged. The next one would be so again, going back to that idea of transient and lingering, what would normally happen, like exercise induced, recovery, a 20-year-old, 25-year-old makes some senescent cells. We look in their muscle tissue 3, 5, 7 days later, they're gone. They've done their job. They've either gone through apoptosis or the immune system's found them. So another thing is that the immune system, most things aren't binary in our body. It's more like, like a continuum. And so what some researchers believe, and this is from modeling it out, is that we would always make some small subset of senescent cells that maybe would be not as easy for the immune system to have found. And so what happens over time is those ones that have escaped detection are now building more and more. So the immune system just can never find them. It couldn't find them when we were 20. It's not going to find them when we're 60. They were always somewhat invisible to it. The third leg would be what's called immune senescence, basically immune system aging. We know our immune system's just less functional as we get old, right? It's less good at detecting and eliminating stress cells, no matter what the cause. And then the fourth part is called secondary senescence, or sometimes paracrine senescence. But it's why often you'll hear a zombie metaphor for senescent cells. Like, part of the zombie metaphor is what I mentioned earlier, right? They're not dead, but they don't make copies, so they're really not living, right? They're just stuck in this between. The other thing is, I had mentioned senescent cells secrete these things into the microenvironment around them. Some of those can take nearby healthy cells and turn them into senescent cells. So that's secondary senescence. And as we age, and And that would be the same on a plant, right? A yellow leaf can attract pests and cause nearby leaves now to yellow as well, right? It can spread that dysfunction. And so that combination of four things ends up creating almost like, think of just a runaway upward slope, right? As we get older, where a little begets more, but then that just ramps faster and faster, like a snowball rolling downhill almost. And so because of that, there's, like left to their own devices, senescent cells after a certain point, that once they become lingering, don't go away. They just gradually accumulate and gradually becomes faster and faster with the passing of time.
I see. So basically, by using senolytic, you are making it slower to accumulate, but because of these different aspects of life, let's say, stress and nutrition and lack of physical activity and all of that, they do contribute still to the accumulation of senescence. And by taking senolytics with that, is that enough? That's, I guess, my question is. Or, I'm on the camp that I'm like, yes, I do think that senolytics may work in ways that to reduce senescent cells, but is that enough if the person is also not looking after the other foundations of their lives?
So it would be, to me, completely different things, right? So, think again of our plant metaphor, right? We still want to make sure that the soil is good. We still want to make sure it gets plenty of water, right? Sunlight, things that the plant needs to thrive, right? Those are the things that you're mentioning, right? Like, it'd be silly not to do those and just like, oh, well, we'll just worry about pruning the yellow leaves. Well, that's not, you're not being a particularly great gardener then, right? So we want to, I think, use senolytics for what they were intended for, right? They're the thing to help, do the equivalent of pruning the yellow leaves, but we want to do our best to make sure that we're not sprouting them all over the place. Right. So,
and you mentioned something very interesting earlier as well. So you said senescence may also occur independent of DNA damage and independent of telomere length. And for those of you who are listening and watching, telomere is this region in our DNA that gets shortened every time our cells replicate. So the theory is that when the telomere length is, minimal and it can't sort of replicate anymore, that's when the cells stop replicating. So you're saying that senescence can occur regardless of that. Is that correct?
And at one point, it was like early 2000s, maybe 2005, they were trying to, this was scientists, they were trying to decide if they should have different names for the senescence that occurs because of telomere attrition, right? And these stress-induced ones, like maybe we should have very specific names. And they
exactly what I'm thinking, because the two different categories.
Yeah, let's just call it all senescent cells, because they have in common this: they don't replicate, they resist apoptosis, they secrete things. Like, they all have these things in common, independent of what caused a cell to become that. But it does cause confusion, because I know many of the podcasts I've been on, the host would think, oh, telomeres, like all senescence is telomeres. And my bias is that relatively little of the senescence we talk about in these studies is because of that. It's more, like I said, 50 replications is a lot.
Yeah.
Right. Like, say, cells in our muscle tissue, I believe an average cell in muscle tissue lives for 7 years, right? Like, does it really need to reproduce itself 50 times? And each offspring can reproduce itself many times, right? And then we still have stem cells as a backup plan to make new cells, right? So, I don't think that is the main cause. I think it's these stress-induced causes that prematurely trigger a cell to go through, and then once it's there, it's kind of stuck. And if it's now making new versions of itself in nearby cells, then then over time, we're just in a game we can't get out of. We've kind of fallen into a rut.
It's like humans, right? Because we can die from old age, just old age in general, or we can die early because we had an accident or a heart attack, or something that induced our death earlier. Not necessarily because our organs stop functioning because of old age, but it's because of something else. So just for a fun fact as well, like you mentioned, our muscle cells average lifespan is 7 years. What are the cells that you know, like the average lifespan of different cells in our tissues?
So I might be off on cells. So liver, it's 1 to 3 years. Closer to 1 when you're super young, like a child. Closer to 2 or 3 adults. Heart cells about 40 years. Fat, I believe, is about 15. Muscle, I think actually I said 7. It might be more close to 10. Some cells are really quick. The cells that line our intestine turn over really quick, right? And then neurons are built to last a lifetime. Like they don't make new copies of themselves. So there are the neurons, and then cells in our eye, like that line parts of our eye, are the only like infinite cells, right, that never make copies. So each tissue would have its own, average lifespan of cells. And I think what you'll often hear is, we replace billions of cells every day, and we replace, like, on average every cell within 7 years, I think, is the most common thing. But that's an average, and it's crazy varied depending on the tissue.
Right. And from the sound of it, we have a lot of cells that live for a long time and can still develop and divide many more times after they reach the end of their lifespan. So theoretically, if we don't have these senescent cells, can humans live way beyond 100 years?
So I would say, just based off animal studies today, what like getting rid of senescent cells doesn't really extend longevity. It makes more of the animals experience, what is their, programmed lifespan, and be more youthful through the tail end of it. So my guess would be, like, senolytics as a category, more at this point their promise isn't like, oh, we're going to make everyone live to 110 or 120. It's let's make it so, like, as an example, common things that you see with aging is that some people by their mid-40s, joints become a real limiting factor, right? Which, that there's an inflammatory component to that usually. And the cells at the get-go that they thought would be most susceptible to becoming senescent, based off early work, were immune cells, and then fibroblasts. And fibroblasts are the connective tissue cell, right? Skin, joints. And that's true, right? That bears out in science. What surprised scientists is that other things, even neurons, can eventually become senescent. So because of that, like, my goal isn't, or, I don't want to imply like, oh, if you do senolytics or do these other things to stave off or repair damage, you're going to live this crazy long time. That's, like, great on you if that happens, but it's the quality of life, right? Like, can you perform better at whatever your age is? And I know for me, that's always been my goal. It's, what does health look like? What can I do to make sure I have more of it now? And what can I invest in to make sure I still have more of it a decade from now, 2 decades from now?
Yeah, it's optimizing the potential that you could have had if you didn't get interrupted by all these external factors to live out to the age that you're programmed to live, in the cells, at least in your DNA. So speaking of which, so what are the hallmarks of aging? Like, we hear about it a lot, on media and podcasts. People talk about hallmarks of aging. I think they're like 6 or 7 or whatever. Is there a standard way of looking at hallmarks of aging?
Yeah, so hallmarks of aging were really more of a model that was proposed in 2013 by a small group of scientists to essentially say these are the characteristics that all organisms seem to share. And it borrowed off what was the hallmarks of cancer, like another model that predates it quite a bit. And so originally they proposed 9. And so senescent cells, or cellular senescence, was 1. Telomere attrition, so shortening telomeres, was another. The genomic instability was a third, but that would be DNA damage is a simple way. But other ones were impaired intracellular signaling, things gone wrong with nutrient sensing, one that's called proteostasis. But a lot of, like, autophagy as an example. Autophagy is largely finding damaged proteins and recycling them. And proteostasis has to do with protein damage. So originally there was this group of 9. And in January of 2023, those same scientists got together and reissued a new paper. And I think it was titled The Hallmarks of Aging: An Expanding Universe, and added 3 to the list. So the 3 new ones was chronic inflammation, which you already talked about, which makes sense. Another one was macroautophagy. So before, autophagy was kind of snuck in to like mitochondrial dysfunction because mitophagy and proteostasis. Now it has its own hallmark, or now it's considered its own hallmark. And then the third one was dysbiosis, so like issues with the gut microbiota as we age. So there's 12 hallmarks, and again, it would be more a model to explain and bundle different aspects rather than think like, oh, this is like absolutely 100% true. Everyone agrees that these are the 12 things. There's, I think, another model is pillars of aging, but a lot of them overlap because the characteristics, most of the longevity science agree. Just how they would then bundle them together, there might be disagreements.
Yeah, fair enough. So you mentioned something about autophagy, and we talked about autophagy every here and there. I want to ask the question directly: does fasting induce autophagy be as does it play the same role as senolytics? Like, is it as powerful as curbing senescent cells as senolytics?
Yeah, so think of autophagy and things or strategies to or the autophagy software program versus the senescent software program as being different things. But they're both stress response. Autophagy just be when stress or damage is fixable still. So things like, as an example, the reason fasting upregulates autophagy is because of nutrient sensing. Cells are constantly trying to figure out what the environment is like around them so that they can respond accordingly. And if there's not enough nutrition, one of the ways that they respond to that is, okay, well, let's figure out what's expendable inside my cell, and we'll use that temporarily for a source of protein or whatever. And so autophagy then rounds up damaged proteins in cells and then recycles it so it can put it back to use. Senescent cells are they're beyond where autophagy is helpful anymore. But the same general idea, like, okay, the environment's poor, we're not getting enough nutrition, what cells might be the best ones that I can sacrifice? And so what things like fasting, or Walter Longo's fasting mimicking diet, his has been studied specifically for both autophagy and immune senescence, so senescent immune cells. And it seems like there's this wisdom built into our cells, both when autophagy, like rather than dismantling useful proteins, it finds the damaged ones. But also when, at a whole cell, we need to recycle a cell. It's the damaged ones or the less useful ones that seem to be sacrificed initially. So there hasn't been a lot of studying on fasting behaviors and senescent cells. It's much more on autophagy. But my bias based on Walter Longo's in his lab's work is that some of these same things, to do the fasting mimicking diet for the immune senescence, usually it's not till 2 or 3 days in that you start to see some of the senescent immune cells cannibalized. So I think, my intuition is that yes, that fasting would be a mechanism to prune senescent cells. And it may be that in some tissues that wouldn't be as useful, right? Useful for immune cells, maybe it wouldn't be as useful in, say, like adipose tissue or fat cells, but we just don't know what we don't know at this point.
Yeah, no, I think that makes sense, and I agree with you. I think, autophagy does occur, but no, it's 48 hours, 24 to 48 hours of fasting, and we don't get to choose what we autophagize, right? We don't get to choose what we're going to recycle, and if the damage is too high, maybe the system itself, the autophagy system itself, may also be dysfunctional, which will lead to us not actually recycling the correct cells. So I mean, all of this is just junctures, just theories at this point, and more studies will be needed to really prove it. So you talked about nutrient sensing, you know, and the very, very important nutrient energy sensing mechanism, mTOR. Let's talk about that for a second. And I'm not even going to ask, is mTOR good for aging? Because I know the answer. It's not a straightforward answer. It's not black and white. I want to ask you, what is the relationship of mTOR to aging?
So mTOR is more specifically a protein sensing, like nutrient sensing pathway. I'm oversimplifying it. And there's more than one variant, right? There's mTOR1, there's mTOR2, and it gets complicated pretty quickly. But I would say one of the unifying things of fasting behaviors is that they're low protein. So like think of like the original, like what I think of as OG longevity work was all on calorie restriction, right? Just we're going to feed animals just, their normal amount of or normal type of food, but much less than that. Make them hungry all the time, right? So inadvertently you're decreasing protein. One of the more well-studied other anti-aging things is methionine restriction. Is that something you've heard of?
No.
Oh. So methionine restriction largely duplicates calorie restriction. All you need to do, like you can let animals eat as much as they want as long as you have methionine below like a threshold, very low amount. It creates many of the same adaptations that calorie restriction does, right? So that would be that's not only protein sensing, it's an amino acid. Methionine is an amino acid for people that might be listening and didn't know. Right, but you can do the same with, like Dave Asprey periodically will talk about doing a 1-day protein fast, right, or a couple-day protein fast. Or we mentioned Valter Longo and his fasting mimicking diet. Well, that's Prolon is the branded version of that people can buy and implement. But the heuristic behind it is to be super low in protein, like calorie restricted, but not only calorie restricted, protein restricted. So all those would then fit into that mTOR nutrient signal, and there are ways that you're trying to get that sensing pathway to say, hey, there's not enough of protein in the environment. And since a lot of what's damaged in cells is either individual proteins or organelles made of proteins, then we recycle those.
Yeah, so that is my general understanding as well, and this is what I struggle with, and please help me with this, right? So the low calorie, low protein in the diet that somehow enhances longevity, right? And then there are also other studies that showed upregulation of mTOR is important for protein synthesis, right? Because it's protein sensing and it helps with anabolism and create and help with recovery. Example, when you finish working out and you have mTOR upregulation, you create proteins to fix the muscles. In elderly, having muscle mass is becoming more and more important because one is to have enough support so that they don't die from fall, because one of the top 3 reasons of death or mortality among elderly is because they fall and then they break something and they puncture the organs. And the other reason is there are more studies that showed the increase in muscle mass, especially in elderly, also showed increase in BDNF and brain-derived neurotrophic factor, and also all these signaling molecules that is helpful with brain health. And as we know, when we age, we are increasing the risk of developing neurodegenerative diseases, and therefore this may also play a big role in living a healthy aging life. What do you think about this? Like, one is mTOR, protein has to be low. The other one is like protein needs to be high so that you have enough muscle mass.
Yeah, so I think it's one of the reasons that Valter Longo's approach appeals to me, right? It's this idea of intermittent, right? Because you're right. We, one of my goals for the listeners, I'm 61, right? So I'm not, I'm at the point where, in theory, it's harder to put on muscle, but I want to go into my next decade with as much as, that allows me to, live my life, right? So I don't want to be a bodybuilder, but I want to, have strong, firm, good muscle quality. So I think there's this tendency to almost oversimplify, like mTOR good, mTOR bad. And I've only ever personally read one circadian rhythm study on mTOR, but what fasting did, depending on when you measured mTOR that day, it decreased it, kept it the same, or increased it, So really what it did is it shifted the rhythm of it, right? It moved the rhythm in time, but the curve was still there. And what I think of, so have you ever, I might have the name wrong. I think it's called it's not the Hooke-Weaver law, but basically it's this old law that goes back to the mid 1800s. But the idea is, think of the analogy for the audience. Think of going into a dark closet. You light one candle, it's like, wow, like it's light in here now. Now you go into that same closet, there's 99 candles lit, you light one more, like, well, who cares, right? You probably didn't even notice the difference. So we have this idea that a quantity of something matters, but what our senses are designed is to measure proportional change. It's why hearing's a decibel system and why that example of light. And my biases receptors on cells are the same, like whether it's the nutrient sensing ones, it's the degree of change that they're measured, they're designed to measure. So intermittently, if we can do that, we don't have to chronically do it, if that makes sense, right? The goal is to create dynamic change so that they'll respond, and then we can go back to normal. We don't have to sustain that change, right? If we sustain it, we're not changing anything anymore. We've just now mitigated that. So like that would be my model. Like, let's do these periodic things that create huge contrast, and then the rest of the time let's work in a completely different way to, optimize, muscle mass, quality, etc.
Right. To sum it all up, what we have talked about is basically, use senolytics, use fasting to reduce mTOR activation intermittently, and then when you're not doing that, put in the resistance workout and really recover well by feeding yourself protein and upregulating mTOR and really making the system work for you instead of you working around the system.
Right. Oh, and I think it's the Weber-Flechtner law is the name of that law.
Yeah, okay. And speaking of, so one of the big thing we do on this show is the takeaway points, right? So what I just summarized is, some of the takeaways we can take from our conversation. But let's talk about senolytics specifically on the dosing, right? What are the optimal doses that someone has to use? What are the different senolytics are there that people can find, people can experiment with? Because I know there are a lot of audience here who like to just buy the stuff and experiment with it, see how they feel. And I always encourage people to do that. And granted, don't experiment with poison, but if there's something that has science backing with it, like take it and feel it for yourself, because you will know your body the best.
Well, I mentioned quercetin already, so that was the first, thing from a plant that was identified. The other thing at the time, dasatinib, is a drug used in, it's immune modulator, so it's used mostly in blood cancers. So that stack from the get-go, and still to this day, is maybe the most studied. And Mayo
and how much quercetin?
In what you'd see on clinicaltrials.gov is usually somewhere between 1 gram and 1,500, depending on the bioavailability of that stacked with it. And part of the reason they stacked it is from the get-go they found that quercetin was active in a certain tissues, dasatinib in other ones, and the combination was just more robust. Then the next thing that I'm aware of in the plant world that was identified was piperlongamine, which is an alkaloid from long peppers, which are a cousin to black pepper. They'd be used in a really similar way. So different than piperine, which is a bioenhancer, but piperlongamine. And then fisetin was the one that got at least some of the longevity people I know excited, because the initial work on that, also done by Mayo and Scripps, was that seemed to be more robust in many different tissues than quercetin was alone, and didn't need to be stacked with something. And so Mayo, I think, either has 9 or 10, last time I looked anyways, on clinicaltrials.gov, ongoing fisetin studies, and the dosing they use is 20 milligrams per kilogram body weight. So, pretty big dose of fisetin alone.
How do you spell fisetin?
Fisetin. So fisetin, some people say as well, which might be correct. I've always just said fisetin.
All these chemical names.
And then, and that's a compound that, like quercetin, would be found in, like strawberries would be a classic example, but just really low amounts. And then the way that they're then usually dosed would be that hit and run approach. So, 2, maybe 3 days, and then take some vacation from them. And the vacations could be, 10 days, 2 weeks. For generically, personally, I wait a month. I just do like the quality senolytic the first weekend of every month. So I do it for those 2 days, and then that's it. I don't worry about it till the next month.
And which ones does Neurohacker Collective sell any senolytics or specifically?
Yeah,
or?
we have a product called Qualia Senolytic, and it has Vicetin. So it would have that milligram per kilogram for about a 150-pound person. It also then stacks some quercetin, piperlongumine, and then things like curcumin, milk thistle, a few other things together that are trying to mechanistically solve some of this resistance to apoptosis in the same way that the original work was done.
Nice. And when we have a code here as well, which is at neurohacker.com/H.V.M.N for our listeners to get 15% off. I'm personally interested in trying for sure. And I don't know if we are going to feel any subjective difference. Do you know if, just trying it once or twice, do you feel any subjective difference in energy, in mood, or any form of biofeedback?
So it really can vary. And like our experience to date is it depends, like one, are senescent cells below that threshold in all your tissues where they're problematic? In which case, pruning some back, you're not going to notice a difference, right? They're below the threshold before, they'll be more below it after, which is, would be my goal personally, but I wouldn't expect to feel anything. So what we did, one of the things we always do, since we tend to make, recipes that have, more than like one ingredient mixed together, often like quasi senolytic, a handful, we'll do our own pilot study first. So not placebo controlled or anything like that, but we'll set out, okay, well, what's a population that would be interesting? Let's, do, like in this case, we recruited people that had joint discomfort, since that's a common area that senescent cells are known to impact. And then we just had them do 3 dosing cycles. And a dosing cycle would be, take it for 2 days, have a vacation, take it for 2 days, have a vacation, take it for 2 days. And then we did before and after then assessment of, we use something called the WOMAC scale, but that's widely used for joint issues. And it measures things like, flexibility, your ability to comfortably do all kinds of activities you would just do in life. And what we saw, and we see this pretty commonly, I don't know if it would be the same for your team, but we tend to bucket super responders, good responders, non-responders. So there's always some subset of people like crazy great response to things. And then, a lot of people just good response. And then there's a small subset typically to anything that just didn't work for them. And so that's what we saw. We saw that with joint comfort, flexibility, things related to that there was a subset of people that they're, if they were on a 1 to 10 scale, with 10 being the worst imaginable, they were like 8, 9s, and 10s to start, and they were, 0s, 1s, and 2s. And then we had quite a few that moved the needle a fair amount. The second study, we did a more general. We didn't recruit with a targeted population. We just recruited people that I think were over 40 and older. And what we saw was, we use SF-36. I don't know if you'd be familiar with it, but it's the most widely used general health questionnaire, and has almost buckets for emotional well-being, general health, physical function, like a range of things. And what we saw was in 5 of the 6 buckets, a change. But the change each person experienced could be different. So what we saw, energy and emotional well-being, were the 2 things that moved the most for the most people, which especially the emotional one surprised me.
That's good to know, so that people who are trying know what to look out for and what to observe, especially how they feel. And one last question: any diet or lifestyle behaviors that you would recommend, related to senescent cells, or even to use in conjunction with qualia, for example?
So I know some people will we'll try to stack taking quiasinolytic with, as an example, maybe like some type of, if they are going to do a periodic fast, maybe they'll do it that day. But I try to do the lower protein personally the days I take it, just to do more to support autophagy as well, since they're both, working on cleaning up damage. But in our small studies and most of the people that we've given it to or customers, we haven't got much. Like most people just do that, and then like they like that they don't have to add other complicated things in. So there wouldn't be anything that I could stand firmly on. I can just say that for me it would make sense. If you are going to do a fasting related thing once a month, like before overlapping with would be when I would incorporate it personally.
Now it's very interesting that there are always two groups of people. One group which likes to stack everything and anything together, and then the other group is like, just give me one thing and I don't have to think about it. And it's very different views on how they want to do their biohacking. So last but not least, if our listeners and audience would like to look out for any information and knowledge and just look for you in general on the social media or online, where can they find you?
So I co-host our Collective Insights podcast, the Neurohacker one, usually more with authors. I'm the one mostly focused on that, but I get some other things. I'm not as active personally on social media, but Instagram is the big channel that Neurohacker is active on, and I put a lot of things their way in terms of what I think would be cool to share. And then I create long blog posts on the neurohacker.com website, especially product related. So if anyone wants to find out more about quiasinolytic, what's in it, why it's in it, why we chose that dose, all of that's in a blog that's called Quiasinolytic, the formulator's view of the senolytic ingredients, something like that. So.
Amazing. Thank you so much. And hopefully I'll be interviewed by you hopefully soon. We're sort of arranging that. And then the tables will have turned, and you'll get to grill me instead.
Yeah, this wasn't a grilling. You had great questions. Thanks.
No, thank you so much for sharing your knowledge and expertise and your wisdom around this. And I've learned a lot today, for sure. And I hope that our audience have also learned quite a fair bit from you. And we look forward to trying it out and see how we feel. All right. Thank you very much.